Welcome back, everybody. In today's video, we're going to be diving into a viewer comment, which is which areas of research do I think are sort of hot topics right now? Things that can maybe have a bright future in terms of securing grants or a position as a PI that could publish, interesting papers. So we're going to dive into that. If you like this kind of content, make sure to give it a thumbs up. It's really going to help the channel grow. It'll show me you guys are interested and I'll be able to keep making videos for you guys. Make sure to subscribe if you do. It's gonna help the channel grow. It's gonna help me know that you guys are interested. And with that, let's get into this. I'm gonna preface this by saying that the ones that I'm talking about today are ones that I've observed to be areas that science is just generally interested in. I'm gonna say that not all of these are things that I strongly believe in, but these are areas that I think have room to grow, and I'll explain what my personal opinions are on each of these. The other thing that I'm going to do is I'm going to let you know that these are more for biological sciences. So if you're somebody that's more into like chemistry or physics, obviously these wouldn't apply to you as much. But I don't know enough about those areas to intelligently discuss those. So keeping in line with more biomedical sciences, what are 3 areas that I think really have a lot of room to grow? The first one that I think really has a lot of room to grow is the microbiome. I think that over the last decade, the microbiome research has really taken off quite a bit, but I still think that in general, this area of research is still in its infancy. I think that the idea of influencing the microbes within our gut and how that can then translate to different metabolic health, disease states, etc., is a really fascinating idea. This is something that has been published a lot and has gotten really high impact publications, including, your Cell Nature Science. I think that there's a lot of funding out there for microbiome, and I think there's a lot of interesting avenues that you could take it that haven't yet been explored. Now, the one drawback that I kind of see to this, and why I personally am not the biggest fan of microbiome research, is that at the moment it seems like everything impacts the microbiome and the microbiome impacts everything. It seems that you could do any little small change and you're going to impact every single phenotype out there within the body. To me, that makes the microbiome not, for one, is easy to understand because there's such subtle differences, can make such large changes. The other thing is that when the phenotype is so striking in that manner, it starts to become less appealing to me. But that's just my personal opinion. I do think that there is quite a bit of thirst out there to learn more about the microbiome. So that's one area that I think really has a chance to flourish. The second is EVs, or extracellular vesicles, particularly exosomes. Now, this is something that for a while I was a little bit skeptical on, but the more I've dug into it, the more that I feel like this is an area that really has a lot of room to run. So EVs could be important for a lot of things. It could be for drug delivery. It could be for how different proteins are delivered to cells for, inter-organ communication, especially in the endocrine field. You talk about microRNAs getting loaded into exosomes and how they could influence distal organs. I mean, that's an entire, huge field all on its own. There's a lot that you could do with exosomes, and I think that exosomes are more than just vesicles released from the cell carrying junk or just random things. I think there is some rhyme or reason why specific things are uploaded into exosomes when others aren't. And this is a field that I think for a lot of reasons, especially for translatability, you talk about how you can hijack the exosome machinery to help, a patient, in a grant application. That's huge, right? That's kind of what you're going for. And especially if you want to get a faculty position at a medical school, that type of research is going to be really interesting to them. And this is something that could easily be carried out in patients because, in humans, it's easy to get a blood draw. And so you could do some good exosome research just from the blood of patients. So I think this is another thing that's interesting. Number 3 on my list has to do with all of the omics. So for a long time, it was RNA-seq. That was like the big transcriptomic data set that everybody wanted. And then they've started to more recently in the last decade or so move into the more mass spec methods. These have become easier, more high throughput, and we're able to get better proteomics, metabolomics, lipidomics. But now they've really started to get fancy with it. You know, you have all of your, of course, single cell sequencing technologies, but you have, you know, not just proteomics, but phosphoproteomics, and you could build a whole phosphoproteome, if you will. These kinds of data sets are huge. And when I say getting into these, what I don't mean is like using it as just one technique. I mean building a laboratory around this type of technique, and building a laboratory that's really based around helping others generate these meaningful data. And for you to be able to get in and develop the mass spec or the sequencing technology to really boost these technologies and move them forward. I think these are extremely powerful technologies that give us so much information, we don't even know how to handle it. I mean, there are obviously packages in R that we use, you know, DESeq2 or, you know, MetaboAnalyst or what have you. But in terms of like really utilizing these, I think we're starting to just get into it now. And I think that these are things that if you can develop the skill set and a new technology in this area, that, you know, you can have a lot of potential in the future. And the last thing, I know I originally said we're going to do 3 things, but I think we're actually going to do 4, is something that I think hasn't been explored quite as much in academia. It's more something in pharmaceuticals, but I think that the academic labs that have done this have really found success, and that's drug delivery. So I'll give you an example of at MIT, there are some labs that work on lipid nanoparticle delivery, which are just these small lipid vesicles that are used to deliver drugs. And this is a really outstanding technology that's been used in pharmaceuticals, and other labs in academia want to collaborate to deliver, you know, anything even to like AAVs, right? So there's a whole bunch of things that you could do with these, you know, lipid nanoparticles or LNPs, but there's in general the idea of how do we deliver different things, whether it's again like an AAV for some kind of in vivo, you know, modification, whether it's some kind of a CRISPR protein, whether it's an siRNA, a biologic, an antibody, any kind of delivery system that you could generate is something that's really valuable and important. And if you could have a lab that's based off of this kind of bioengineering, I think this is something that can really have a bright future. So those are 4 areas that I think for the most part have a leg up at the moment. Not to say that anything else doesn't. I think that, you know, you could do anything. You could do something in virology, you could do something in embryology. You know, there's all kinds of fields out there. They all have their place. They all have the ability to publish high quality work in good journals, you know, for you to be able to get funding, for you to be able to find a faculty position. These are just 4 that off the cuff, in my experience from what I've seen, are ones that have the slight advantage in terms of what people are looking to get right now. But that all could change. So for example, the COVID-19 pandemic, I think really changed the way that we're going to look at science. And I think that infectious disease research and vaccine research is going to get a huge boost. Those areas, you know, maybe didn't quite as much have that boost before, but now I think a lot of funding is going to get funneled into those efforts. So these things are all fluid and dynamic, and they could always change. So, you know, always be on the lookout for the next best thing, but also keep in mind that these things do change, you know, year to year. They're not always going to be the same. So with that, I'll catch you guys in the next one. Bye.